Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsors, Ncardia and SCIA Biosciences, I'd like to welcome you to Rethinking Translational Predictivity using hiPSC-derived models, a retrospective analysis for cardiac liabilities. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now I'd like to introduce our presenters for today. First is Dr. Greg Luhrmann, Ph.D., Technical Director, North America for Ncardia. Our second presenter is Dr. Xiaoyu Zhang, Ph.D., Senior Scientist, R&D Applications with ASIA Biosciences. And our final presenter is Jean-Pierre Valentin, Ph.D., Senior Director and Head of Investigative Toxicology at UCB Pharma SPRL. So with that, I will welcome our first speaker and we'll get started. Welcome, Greg. The presenter ball is yours.
Thank you very much, Elizabeth. Okay, thank you everybody for joining us today, whether you're calling in from Europe or Americas or wherever you are. I just wanted to give you a quick overview of the discussions today. I'll be giving a, not a brief, but a pretty extended introduction into current cardiac safety testing practices, basically stemming from what we do well to maybe what we don't do so well, and seeing where we can clean up on some of the failures that we've had over the years and improve cardiac safety testing going forward with the use of some of these emerging stem cell derived cardiomyocyte technologies, as well as implementing these with some of the instrumentation out there, more specifically with our friends from ASIA using their Accelligence technology. So Xiaoyu will, from ASIA, will give a brief introduction to the technology after I'm done. And then Jean-Pierre Valentin will demonstrate sort of a case study from how UCB has used these combined technologies to provide them a more comprehensive review of some internal compounds that they had in development. So, as you're probably aware, there have been a number of costly, high-profile drug-induced cardiac deaths that really came to light in the 1990s and early 2000s. Many of these deaths were actually found to be from a type of lethal cardiac arrhythmia known as Torsades-de-Pointes. And these sort of high-profile cases really provided the rationale that helped uncover the role of drug-induced block of the cardiac potassium channel known as hERG. This identification of hERG actually led, as the culprit behind a lot of these cardiac arrhythmias, actually led harmonizing agencies around the world to adopt guidelines which require preclinical and even clinical studies to determine basically the potency of hERG block in the form, I guess, actually determining the delay of ventricular repolarization as it's laid out, as well as the risk of clinical QT interval prolongation. Since the implementation of these guidelines, these are known as ICH-S7B and E14, the industry has really not had any drugs pulled from the marketplace due to these lethal arrhythmias. So this has been really a major win for the industry, seeing as how we've now adopted these guidelines based upon sort of data that came to light that really has moved the drug discovery industry forward and helped to prevent a lot of these deaths. But it must be noted, I think, that that's a lot of this focus has been on hERG block, and this sort of acute ion channel hERG block is really only a small piece of the cardiac safety puzzle. There are a number of different mechanisms at play which can result in even life-threatening cardiac adverse events outside of acute ion channel block. So some of these modes of cardiac toxicity are laid out pretty clearly here and can largely be grouped into three major categories. One, functional cardiotoxicities, structural cardiotox, and sort of indirect effects, maybe those which have more effect on blood pressure related mechanisms. So add into this these sort of ever-evolving patient genetic risk and comorbidities associated with patients, that may even include drug-drug interactions that we just don't see during preclinical development. So all these sort of risk factors can actually exacerbate a number of these molecular mechanisms, ultimately ending up in heart failure, cardiac disease of some sort. A lot of focus has been put into knowledge about QT prolongation and Hurg block, but now we start to see that this entire picture is a very extremely complex organ, this heart. And there's a number of different failure modes that can be at play here. So this has really necessitated a much more comprehensive cardiovascular safety assessment paradigm that has actually historically relied upon a number of different animal studies to pick up these various cardiac issues. So currently the CV safety funnel is basically some variation of what we see here at the majority of companies that we interact with. So early on, we see some, ion channel evaluation that's at a minimum Hurg, if not other ion channels as well, calcium, sodium, etc. These are typically followed by a few ex vivo and in vivo early tox studies. Prior to entering into these clinical trials. So, of course, as it denotes here, throughput and the number of compounds proceeding through these gatekeepers or these filters are greatly reduced as it progresses through these different studies. So if the stars align properly, only a few of these compounds will actually proceed through to lead optimization and ultimately clinical trials. So this, the resultant clinical trials is, of course, not the end of the story either. As we know, a majority of compounds will still fail in safety trials and even post-marketing for a number of reasons, most of which is primarily for cardiac toxicity reasons. And I'll kind of go into data here in a little bit showing you evidence of that. So to this point, a 2014 publication concluded that the absence of toxicity in animals actually has low predictivity for the lack of adverse events in humans. So that means just because we don't see an issue in animals, that doesn't mean that we're not going to see an issue in the clinic. On the other hand, we're actually pretty good at picking up these various cardiotoxicities in our animal models. And these actually correlate quite well with identifying adverse events in humans. So if we know a drug is toxic in an animal model, it's pretty much most likely going to be toxic to humans as well. So that's a very important piece of the puzzle to keep in mind as we proceed here, is that our animal studies are actually pretty good. So interestingly, to this point again, some of the most concordant observations, namely QT prolongation and detection of arrhythmias, are perhaps our most predictive assays, is kind of what this paper concluded. So maybe that's why we actually put so much focus on understanding the mechanisms behind these arrhythmias, is because we're actually really good at it, and the animal models are actually pretty good at detecting these acute ion channel blocks leading to arrhythmias. But this brings me to the point that apparently we're still missing a lot. Even though we have a pretty good handle on some of these animal models and their translation to the clinic, we're still missing a lot. So I wanted to go over some of the key failure modes that we can also see in addition to this acute Hurg issue, with the subtext to keep in the back of your head that we are typically actually very often knowledgeable of QT issues going into the clinic. It's just sort of a risk-benefit analysis that we have to do in the back of our heads at all times. So keep that in mind as we go on. So compounds tend to have a tendency for instigating cardiac adverse events. I know when I was working on a preclinical team at Pfizer, the drugs we were working on, though they were neuroactive drugs, they actually had cardiac adverse effects as well. So basically through all stages, what this slide from Will Redfern demonstrates is that through all the stages of a compound's life cycle, cardiovascular issues are basically at the top of the failure list at every point, even in post-marketing. So even in preclinical studies, cardiovascular issues still account for a quarter, even up to 40% of the identified issues that we see, depending on the company or the publication that's out there. And this would all be okay if we are identifying these issues early and filtering them out during your preclinical studies. And if that actually solved the problem, that'd be awesome. But guess what? We still see, even after implementation of ICH, SFB, et cetera, we're still seeing about a quarter, typically a quarter of the clinical failures are still due to CV side effects. So there's a number of different mechanisms at play here, but maybe this actually these existing toxicities in these humans, maybe this actually points back to my earlier statement about having to account for all these patient comorbidities, drug-drug interactions, and genetic factors, which we just cannot model in our animal models here. So kind of switching gears here, I think really a big problem is where and when these effects are being picked up. So as you can imagine, if a drug is failing in a late-stage clinical trial or post-approval, R&D costs are already tens of millions, hundreds of millions. Conversely, if your project team has advanced a compound or two through lead ops, or maybe you're a small biotech with only a handful of chemical matter, to your company's name, you may still try to push the limits of your safety margins because, for a number of reasons, maybe you have real, great target potency. Maybe there's a patient population that you're trying to reach. But at this point, you've invested years of development and millions of dollars or euros. You can't go back and retool your compound if a safety issue pops up too late. So it's always something that we at Ncardia are pushing, is to try and get the adaptation of more predictive assays much earlier. So at this point, it becomes a balancing act of sorts, weighing human lives against potentially life-saving treatments, or, as I just mentioned, sort of weighing our careers of pushing successful compounds into the clinic versus failures. So as I mentioned before, in many cases, we're actually cognizant of the risks, not just going into the market, but even into early clinical trials. I think this has really never been more evident than what we see during the sort of R&D and production of oncology drugs. So this is actually, this publication here actually provides a spectacular review that details an exhaustive list of both preclinical and clinical cardiac events that are both observed with various approved kinase inhibitors on the market for oncology purposes. So though I'm just showing the first 6 drugs here from this really pretty exhaustive list, this report clearly demonstrates that we enter into clinical studies with prior knowledge of a plethora of cardiac issues. And what was actually reported are, the major issues are typically QT prolongation issues and structural cardiotoxicity injuries or events. These are oncologics. A lot of this may actually be due to the primary pharmacology or sort of on-target toxicity. But we've actually seen a number of next-generation kinase inhibitors hitting the market with much lower cardiac toxicities or liabilities than their predecessors. So we realize that there are issues out there, and we're trying to make improvements. But we're still entering in a lot of these clinical trials, knowing that there are major issues that will pop up down the line. So there are a number of supporting literature out there that confirm these structural cardiovascular issues. These often result in cardiac arrhythmias. This isn't just acute HERG block. This is other issues that may ultimately result in arrhythmias. And there are a number of different mechanisms at play here leading to heart failure that we're just not picking up in our current safety screening practices. So some compound effects, may just not translate in our animal models. Maybe they don't have the potency seen in human tissues, off-target effects that we see in human tissues. Animals may be dose limited in many studies. Maybe we're only designing studies for more acute toxic effects. So, there's a number of things that we're likely missing here, and ultimately this points to a need for more predictive and comprehensive strategies for cardiovascular risk assessment. So, emerging technologies, are these going to offer a new way forward for us? And of course, the answer is, is going to be yes, of course, or otherwise I wouldn't be giving this talk. So I just wanted to show a quick slide here. This is a recent survey from the Safety Pharmacology Society that points out a couple of different things here. One, that human models are actually now considered more relevant than a lot of the animal models out there. Whereas actually, we may have mixed feelings about the predictivity of our animal models that we so highly regard and so highly rely upon. So in reality, I think this is maybe not well founded. I think I've pointed out that actually our animal studies are actually pretty predictive of toxicological measures. The issue is that we're still just missing a lot that we're not picking up in these animal studies. So maybe the use of more physiologically relevant models, i.e., human tissues, would likely benefit us in the long run. So maybe the secondary issue here is the sad but true point here is that safety pharmacologists maybe don't even really consider reducing animal use as a major concern, and that's kind of sad to me. Just a side note there. So switching gears, just as these harmonizing agencies, the FDA and academics and the pharmaceutical industry came together, a couple decades ago to push the formation of ICH7B and E14, we now have those same bodies at work, which are pushing the next generation of our mechanistic understanding of cardiac safety issues. So you may have heard a lot about the SIPA initiative. This is the Comprehensive In Vitro Proarrhythmia Assay. I'm not going to go into any detail whatsoever on this, other than to let you know that we've developed a very good mechanistic understanding of how ion channels play together, and then how we can actually pick up these arrhythmia effects in these cardiac stem cells. Cardiomyocytes derived from stem cells. But again, I remind you that this ion channel pharmacology is only a very, very small piece of the pie. We still need to develop a mechanistic understanding of all these other mechanisms at play. Perhaps more importantly, understanding the structural cardiotoxicity events that may be popping up and the mechanisms behind that. So a recent meeting last month, sponsored by HESI, has actually focused on just that. And that is, going beyond SIPA and now focusing on some other issues. So what HESI is doing is actually creating a dialogue amongst the stakeholders in the field to help further our understanding of these other mechanisms that may be at play leading to cardiotoxicity, namely contractility and structural cardiotoxicity. So to date, really just briefly wrapping up how the FDA views the SIPA-like studies, they actually are thinking of using these SIPA studies much later in the R&D pipeline. I think this idea has largely been developed under a very limited scope, i.e. acute hERG block, that it's often associated with these clinical arrhythmias. But we believe that using these stem cell cardiomyocytes at this very late stage is very limiting. At this point, you've already spent a lot of time, money, and animals, for that matter, on your candidates. We really see the role of cardiomyocytes and technologies like ASEA has here as being useful much earlier in this process. We actually see that using efficient screening strategies, which can even address chronic and structural cardiotox issues, in addition to the acute ion channel issues that we've previously been looking at, these screening strategies we found can actually be quite predictive and very easy to use on these impedance and MEA systems that our friends at ASEA actually produce. So the use of these assays now, which can help aid like chemotype selection and SAR campaigns, actually allows us to guide medicinal chemistry efforts to design out Cardio.Tox effects much earlier. We can sort of remove the toxic chemistry while maintaining efficacy much earlier with these early project teams. So we've actually seen a number of our customers using this sort of paradigm to guide the early preclinical development. And I'm just really briefly going to fly through these. I'd love for you to come back and look at some of these publications that I'll mention here in much greater detail. But basically, these are publications which use this sort of veritable catch-all cardiotox screening program. And that is the combination of these iPS-derived cardiomyocytes with the ASEA Excelligence technology. And I'll let Xiaoyu introduce that technology in a lot more depth than I can in just a minute. But yeah, I just wanted to point you quickly to some of the studies from the literature where pharmaceutical companies have used these in their early R&D process. So one here is by a group from Janssen where they've looked at HDAC inhibitors and the cardiotoxic effects of those. Additionally, we've seen work from the FDA specifically, outside of SIPA even, looking at more structural cardiotoxicity assessment. Here they used Excelligence technology to enlighten themselves about doxorubicin-induced cardiotoxicity. This is being mediated through upregulation of death receptor pathways. And then finally here's a great publication from Boehringer Ingelheim, which actually paid specific attention to the various structural cardiotoxicities and other cardiac failure modes mentioned earlier. I really urge you to read this paper in full as it details how Boehringer implemented this assay into their early project teams to de-risk cardiovascular issues prior to going into their animal studies. So finally, I just wanted to summarize by saying that we at Ncardia are doing a lot of work to correlate the results that we see on the Excelligence Cardio to troponin, as you're probably familiar, is a very commonly used clinical biomarker of cardiotoxicity. Long story short, we see really good correlation with the data coming out of the ASEA system, correlating nicely with troponin release to give a reliable marker of cardiotoxic injury. So with that, I just wanted to finish by quickly running through Ncardia's capabilities. Not only do we produce these stem cell cardiomyocytes, fibroblasts, and related technologies in the cardiac world, we also offer a number of safety and tox services and efficacy screening services, in addition to our pipeline of cardiac-focused and neuro-focused products here. And then I just wanted to throw out that we also offer what we call cardio plates, and these are basically cells which are pre-plated on a number of different plate formats, including some of these Excelligence plates as well. So with that being said, I wanted to wrap up and pass the ball over to Xiaoyu from ASEA, where she could provide a much more detailed introduction to the Excelligence technology.
Thank you very much, Greg, for providing us such comprehensive background about cardiac field and talking about what we are actually missing for cardiac safety evaluation. In the interest of saving time, I will give you a very short introduction about our Excelligence RTC cardio ECR system from a technical perspective. Even though I won't be able to show you our own cardio ECR data, but I do hope my slides will help you to know the features and potentials of cardio ECR system, which will guide you to utilize the system for your own cardiac safety evaluation, drug discovery, and a cardiac disease model study. So in my talk, I will give you a very brief introduction about our company and which followed by an overview of cardio ECR system. So our company, ASIA Biosciences, was founded in 2002. Our headquarters are located in San Diego, California. Now we have more than 500 employees. We do direct sales and support in US. In the meanwhile, we have more than 30 distributors globally. So this is our cardio ECR system. As you can see, the system has 4 different components. This is the control units, which is the laptop and the analyzer and the plate reader. And this is our specialized plate called cardio ECR plate. One of these advantage of the system is that the plate reader is actually placed in the incubator permanently, which allows the user to conduct both short-term and long-term compound treatment without any human involvement. And most importantly, the cell response to the treatment can be detected under the physiological relevant environment during the entire experiment. Now let's look at the plate. So as you can see, there are 2 sets of electrodes, impedance electrodes and 2 field potential electrodes in the middle. Due to the separation of these 2 types of electrodes, cardio ECR system is the only platform allowing for simultaneous measurement of impedance and field potential signals from cardiomyocytes. Now you may ask how the system measures the impedance and what the impedance can do for us. So on these slides, I will explain how the system detects impedance signal and why we can use impedance to evaluate cell viability and measure cardiomyocyte contraction. So as you can see, these are impedance microelectrode arrays on the bottom of each valve, which are working as one big negative and one big positive terminal, which measures the current floating between electrodes from the entire cell monolayer. So in order to generate impedance signal during each recording, the systems actually apply very low voltage, which is about 10 millivolts to the plate. If the walls of the plate only contains cell culture medium, there will be very low currents flowing between electrodes. Once the cells are seated in the plate and start to attach the bottom of the wall, they will act as resistors to impede currents. Therefore, the impedance is actually affected by the cell number. So more cells, higher impedance, and also the cell attachment, stronger attachment, higher impedance. In addition, different morphology of the cells will generate different impedance values. For the beating cardiomyocytes, there will be changes in both attachment and morphology during each contraction. In order to capture these very temporal changes during the contraction, the data acquisition rate of our cardio ECR system is actually 2 milliseconds for the entire plate. So in these summary slides, you can see what information you will gain from each valve. First, you can actually evaluate cardiomyocyte contraction and variability via these impedance electrodes. And at the same time, you can actually evaluate integrated ion channel activities of cells by using these ECR electrodes via this field potential readout. So besides these simultaneous recording of impedance and the field potential signals, the system also provides a built-in electrical pacing function via these impedance electrodes. Due to the direct contact with the cells to the electrodes and the large well coverage of these electrodes, we can use much lower power to pace the cells compared to the other conventional pacing methods. Therefore, you can perform long-term electrical pacing without harming the cells. At least from our data, we haven't observed impairment of cardiomyocytes due to the long-term pacing. The highlights of the system are label-free. As you can see, no dyes are required for contraction measurement and the cell variability evaluation. it shows very high predictivity when using iPSC-derived cardiomyocytes to assess drug pro-arrhythmia liability. The system also can evaluate cell variability, contraction, and electrical activity from the same well at the same time. Last but not least, the system are suitable for both short-term and long-term assessment of compounds. The major application of the system first, you can use the system to evaluate cardiac toxicity, which you will see more data from our next speaker. In addition, the system can actually provide you functional readouts of your iPS-derived disease models. So you can use the system to study your cardiovascular disease cells. And these days, we actually see more and more companies start to use this system for drug discovery, especially for the drug affecting cardiac contractility. Even using the cardio-ECR system to measure the contractility of the cardiomyocytes is not in the scope of this webinar. I still want to quickly show you some of the data. So impedance-based contractility assay has been well validated by multiple groups. The beating amplitude, as shown on this impedance trace, is a surrogate for the force of contraction. The compounds reducing force of contraction, called negative inotropic compounds, do decrease beating amplitude. However, due to the immaturity of iPSC-derived cardiomyocytes for most positive inotropic compounds, including isoproterenol, they didn't show positive impact on the contraction of iPSC-derived cardiomyocytes as shown in this publication. So we actually test a few positive inotropic compounds in pluricytes from Ncardia. Gladly, we actually see a big increase in beating amplitude after isoproterenol treatment. However, if you look at these two positive inotropic compounds, we didn't see the beating amplitude increase after the treatment. But after we applied long-term pacing, now we start seeing the increase in beating amplitude after these two positive inotropic compound treatment. So therefore, this data suggests the long-term pacing using cardio-ECR system can improve maturation or functional status of iPSC-derived cardiomyocyte. Thank you very much for your attention. Please let me know if you have any questions. Now I would like to pass it to our next speaker, Dr. Valentin from UCB. Thank you.
It's a pleasure to be here with you this afternoon or this morning, depending where you are. So the purpose of this presentation is to share with you some experiments we conducted some time ago regarding the assessment of drug effect on electrophysiology, contractility, and viability in human-derived pluripotent stem cells, cardiomyocytes, using the RTCA cardio-ECR platform that has just been presented. I would like at this stage to acknowledge the core contributors of these studies, Annie Delanois from UCB and Tessa Decourt from Ncardia. So again, very briefly, because it has been nicely introduced by Greg, the background is, as we all know, drug-induced cardiac toxicity is still a challenge in drug discovery and drug development, not only around the electrophysiological aspect, but also the structural cardiotoxicities, as well as the indirect effect. And this has a knock-on impact on attrition, but also delays in drug development and impact on approvability and post-approval requirement. And raising a number of questions in terms of developing novel, better, more predictive assay, enabling to form an integrated risk assessment that can be used for internal decision making. So the study that I will be sharing with you in the next few slides will simply assist in the ongoing debate around the value of using functional data generated from iPSCs derived cardiac myocytes for predicting cardiotoxicity in a clinical setting, and how this model might improve or help us making better, more informed decisions early in drug discovery and drug development. So the aim of this retrospective study was to test the effect of 3 UCB proprietary compounds, which were discontinued in preclinical or clinical stages due primarily to cardiotoxicity. And we looked at the electrophysiological aspect and impedance of human-derived cardiac myocyte using the Excelligent system as presented earlier. We look at both very acute as well as delayed effect of increasing concentration of each of these compounds. I will just give you again very quickly, because it was nicely presented earlier, give you some background of the system that we use. In this case, it was the Excelligent RTCA Cardio ECR instrument, which enables the simultaneous recording of cellular impedance presented in and summarized in this presentation as cell index, and the electrical field potential presented as extracellular recording. So the extracellular recording measurement captures the changes in the extracellular field potential generated by the electrophysiological processes across the cell membrane. On the other hand, the impedance measurement serves as a surrogate marker for contraction by measuring the alteration in impedance when the cardiac myocyte monolayers beats together. So in addition, it can be used to study the overall cell viability over time, and that's the context where how we have used these parameters. So what about the experimental design we follow for this study? So we use hiPSC-derived cardiomyocyte from Ncardia, the pleurocyte aspects. Cells were cultured for 8 days to reach confluent and steady state on a 48 wells E-plated and daily monitored using the Cardio ECR system. The compound were added and parameters recorded for 24 hours after day 8. So you can see the set of concentrations that were used, and the concentration were selected to cover those that lead to cardiac toxicities in a clinical setting for compound A, and in non-clinical assay for all 3 compounds. So again, you have seen this in terms of data collection. Each condition was present in 3 wells, and for each wells we were measuring, as mentioned earlier, 1 electrode was associated with a measurement of impedance, and 2 electrodes were measuring the field potential. And you can see here on the bottom part of the slide typical traces, typical recording with the cell index and impedance changes, as well as the 2 field potential measurement. And this was recorded here over 60 seconds. So you can see that just eyeballing the sweep, that overall the bit rate was in the region of 20 to 30 bits per minute. So what are the parameters that were extracted from such a recording? So on one hand, the impedance at the top part, so the bit rate was calculated, as well as the incidence peak that you can see here, the peak amplitude, and the variability overall over time. In terms of the field potential at the bottom of the screen, the bit rate could obviously be calculated as well as the spike amplitude and the field potential duration, reflecting in some way the action potential duration measured in a kind of more traditional action potential assay. And these different components reflecting, as I said, the action potential shape and size with the depolarization aspect and the repolarization component. So the data were analyzed at different time points, because we have to acknowledge that one of the challenge currently is the capacity to analyze this huge amount of data in a sequential way. So the data were, as I said, recorded over 24 hours period and analyzed at different specific time points across a range of concentrations, starting with the vehicle presented in black in all traces, As you can see on this slide, for all of the 3 compound investigative, the profile of the vehicle effect was quite consistent. And just some key messages for each of the 3 compounds. So compound A, you can see an acute decrease in cell index at the highest concentration tested, and overall concentration dependent decrease in cell index at all the other concentration in terms of delayed effect. In relation to the second compound, UCB, an acute increase this time in cell index at the top concentration, and overall concentration dependent decrease in cell index at a later stage. Compound C overall show no effect in an acute setting, whereas there were some decreases in cell index at all concentration in terms of delayed effect. So from what you can see from this slide is we could distinguish with this compound 2 phases, an acute effect versus a delayed effect, acute effect being within the first 30 minutes and delay being beyond that point. So these drivers, in a way, we decided to further analyze the slide. But before that, moving to the second aspect, which is the field potential. So presented here are a sweep of 60 seconds extracted at different time points at baseline, 10 minutes, 4 hours, and 24 hours after compound administration. What you could see here, again, that the vehicle treated sweeps were very consistent, very reproducible over the 24 hours period. And you could see that at the 1 micromolar concentration, which is the green band here, you could see reduction in bit rate at all time points, so duration 24 hours and over. Regarding the other concentrations, 3, 10, and 30 micromolar, the cell showed some irregular beating shortly after compound administration, which continued for at least 24 hours. So we could again see here different effect depending the concentration assessed. So as I said, one of the challenge was to analyze this huge amount of data in a way that would be meaningful for us in drug discovery and to help us making some clear decision and recommendation for each of these compounds. So here presented are the kind of time and concentration dependent effect plotted over the 24 hours. The data here are normalized to a baseline value as being 1. And the time course is presented for each of the parameters. So top right-hand corner, the changes in spike amplitude. At the middle of the graph, the changes in bitrate calculated using the 2 different approaches, and at the bottom, the change in field duration potential and the cell index amplitude. As you can see, this enables to have a quick understanding of what's happening over time, but it's probably insufficient to help us making some high-level integration. So what we decided to do is to look at another mean to analyze the data and to integrate this rich information. And we look at the area under the curve calculated for each parameter at each concentration. From that, we were able to generate this kind of what we call a spidergram type graph with focusing on the 4 key parameters. So the beating rate, the field potential duration, the spike amplitude, and the cell index amplitude. The baseline value being plotted here at 1. And as you can see in this case, if we just focus on the left-hand side, the acute effect over the first 30 minutes, there was a decrease in the beating rate at concentration above 3 micromolar, a decrease in field potential duration, as you can see here, concentration dependent decrease in spike amplitude, highlighted here, and a biphasic effect in terms of cell index with an initial increase at low concentration and a further reduction at higher concentration. So you can start to visualize the effect. And again, in these instances, we divided the 24 hours time scale in two windows based on the initial assessment. The same analysis was subsequently conducted for the other two compounds, and you can start to see some differences in profile. In this case, it's also worth highlighting that some irregular beating were observed from 3 micromolar onward, type of arrhythmia-like event observed at the highest concentration tested. And consequently, there were no field potential duration detected for a significant period of time at the highest concentration. I am also now moving on to the third and last compound that was evaluated. So less dramatic effect, but nevertheless some significant effects were observed. And during the first 4 hours after addition at the top concentration, which was here 10 micromolar, some arrhythmia-like beating pattern were clearly detected. So as you can see from this overview, it's possible to get a good indication of what the time course of effect are for these three compounds. However, this visualization does not enable to compare and to rank compounds between each other. So therefore, we plotted the graph on the same slide in order to be able to really look and compare across the different compounds. This was not the objective of this study. We were more interested by the potential mechanism and translation and correspondence to existing model, but this is certainly an option that can be used to rank and compound in a drug discovery setting. Again, presented here are just the period between 30 minutes and 24 hours, but all different time points could be plotted. Then the next question, and that was the overall objective of this retrospective study, was how do the data generated in this test system compare to the data that were generated previously with this compound in more traditional and well-established models. So presented on this slide are a summary, a written summary, kind of semi-quantitative assessment of the effect of these three compounds on the different parameters, and the different color code correspond at changes, and we will look in the next slide how they relate or not to changes observed in more well-established models. So now if we look at the effect of these three compounds in other models, as you can see, these compounds have been tested in a number of models. Why? Because these compounds, some of them were in preclinical development a number of years ago, and some models were applied to these compounds prospectively. So as you can see, presented here are the effect on cardiac ion channels such as sodium and calcium with different level of potencies. Compounds were also evaluated in the Purkinje fiber assay, in the zebrafish as well, in the guinea pig and non-rodent telemetry. Typically, it was a dog study, and also one compound was evaluated in clinical setting in single ascending dose studies in healthy volunteers with the effect that you can read here. So again, just trying to relate this well-established model to the cardiac myocyte, can see that a number of changes were able to be picked up with some kind of translatability. Let's pick up some, for example. So a decrease in spike amplitude, which may relate to the decrease in Vmax in the Purkinje fiber. a reduction in the field potential duration corresponding to a decrease in the duration of the action potential in the Purkinje fiber, a decrease in beating rate corresponding to a decrease in beating rate in the zebrafish, increase also in beating rate at kind of higher concentration, again that was identified and detected in the guinea pig, some irregular beating and some beating arrest, and again referring here to the clinical observation of syncope and AV block. And you could do a very similar analysis across the three compounds. Just again to pick up some other example, an increase in field potential duration associated with increase in APD and increase in QTC interval. So overall, some good, at least qualitative translatability across model. Moving on to the follow-up slide, because obviously these studies did not give us all the answers we were necessarily looking for. But I think going beyond this study, I think we could envision using this model in a wide range of contexts. One option could be, again, depending the question being asked, to quantify the reversibility of the effect, which is something we didn't do. Look for other biomarker of cardiac injury, such as troponin, microRNA, and others. Refining the concentration range, because our concentration range was quite wide, half log or 1 log unit increment. Looking for effect on cell metabolism using other approaches, using a drug-drug interaction, in particular for certain diseases where patients are expected to be receiving different medication, and this could be explored in such a test system. Looking at repeated administration over a period of several days could also be explored, and I'm sure there may be many more options available to the scientists. We could also obviously look at other in vitro test systems to address specific questions. So, conclusion from this initial pilot studies, I think we concluded that the hiPSC-derived cardiomyocyte in combination with the EMA impedance assay technology could capture some relevant cardiovascular effect of our proprietary compound that were in effect previously detected using more traditional models. We believe also that this approach could be, that cardiomyocyte could be a useful model to generate predictive data on multiple parameters, therefore avoiding to use several models independently. Obviously, we have to be also open, honest, and transparent that this model may not detect at this moment in time everything, and this may be linked to the maturity of the cell, for example, and I'm sure all the providers are working hard to improve constantly their model. So I will probably stop there, looking at the time, and be happy to take any question.
Okay, thanks, Jean-Pierre. I'm going to sort of handle the host duties here. We've got a couple questions that have come in, the first of which may be related to the introduction. So what is known about QT shortening and the relevance? So let's see, it says, yeah, QT prolongation is a well-described surrogate as a risk factor for arrhythmia. What is known about QT shortening and discuss with respect to preclinical and experimental studies? Maybe this is a question. I'll maybe take first crack at it, and then Jean-Pierre, if you have any insight, let me know. So to my knowledge, QT shortening is not really a major issue. It's definitely not something that pops up a lot, and I don't think the FDA is really too concerned with it. I know that there are short QT syndrome, just like there's long QT syndromes. So like familial QT shortening that's often actually associated with risk of sudden death and ventricular arrhythmias, similar to what we see with long QT syndrome. So I think any data I've seen is well, there's not a lot of data. There's only been, a couple of drugs that I've seen out there with any QT, major QT shortening effects. So Jean-Pierre, I don't know if you guys stumble across that at all or not.
Just to complement your answer, I think a survey was conducted under the auspice of the Safety Pharmacology Society a few years ago, looking at whether this was an issue, a real issue in drug discovery. And the overwhelming answers were that it was not an issue from a drug discovery point of view, because as you said, only very few compounds do demonstrate this ability to shorten the QT interval.
Mm-hmm. Yeah, but in theory, those would be picked up in these sorts of assays as well. So, okay, I know we're up against the hour mark here, but for those that want to continue on, we had just a couple more questions from the audience. So one of these is, let's see, to Jean-Pierre. Thanks for sharing the data on failed compounds, as mostly pharma only show the successes, even though we know that there are many dozens of failures in preclinical studies every year. So is it clear from the data where to set the cutoff points? In other words, how much of an effect and at what concentration would trigger a decision to discontinue a candidate? Jean-Pierre, any insight on that?
Well, I think it's a kind of $1 million question, and it's a very pertinent question. I think we have some confidence that we've gained over the last 10, 15 years to define some criteria and some cutoff for certain assays, ERG being probably the best example, with all the literature supporting both in terms of absolute value as well as kind of safety margin or separation between efficacy and safety. So we have a good understanding for certain models. I am personally not yet convinced that we have reached, as an industry, that level of confidence for some of the more novel assays. And what was presented today were some of these. I think as we learn more of this, running more reference compound, and doing more retrospective studies, and several have been published that were presented in the introduction, I believe we will be able to confidently define some threshold and establish some margin. But I would say at this moment in time, it's probably too premature, at least for me. But some other colleagues in the industry who are using these assays may have gained this level of confidence already.
Thank you. We just had another question pop in. How do you really use the CI, the cellular index, or amplitude of effects for making go, no-go decisions, since it is mainly an index of toxicity or viability of the cells? Jean-Pierre, I don't know if you've had enough experience with this yet. I can comment afterwards. I think we've got some idea. But how do you guys use this cell index value?
Yes, you are probably better placed than us to answer this question. I think it is fair to say that the experience that we are gaining is challenging. Effectively, we are using it in terms of cell viability. That's clearly a very good index. The other indices are there and being considered as an integrated risk assessment at the first glance, and together with a more well-characterized assay. Greg?
Yeah, I would echo that. The cell index value is only sort of one measure, and that it provides a sort of a limited view as to what's going on. I think it's important that we understand how that fits into the other signals that we're getting, even within the same system. I think, in my experience, we've screened a lot of compounds, internally and for external customers, and we see the cell index value as being a pretty predictive measure of even structural cardiotoxicity. I can even point to that Boehringer-Ingelheim publication, which was looking at compounds that failed due to a number of cardiac structural issues. And these were basically being picked up at very, very similar, typically within a half log of the concentrations that we're seeing these structural cardiotox issues in anesthetized dog models. These were being picked up as part of the cell index function. So I think that you can run a number of cell viability assays, but it's really these cardiomyocyte, these stem cell derived cardiomyocytes, which are seemingly much more sensitive than sort of sort of the standard tox models, standard just cell viability models out there. So using these integrated systems in addition to the beating rates or the field potential duration changes, like it's an integrated system that you have to sort of all take into account. So it's kind of a difficult question to answer. So
just perhaps a point to add to this. I think as you mentioned, Greg, in your introduction, I think there are many cardiotoxicities out there that we are struggling with in the pharma industry, and having these integrated models together with the associated relevant technology may enable us to get a better feel, a better understanding of the toxicity, cardiotoxicity of our drug candidates.
So I think we need to wrap this up. I see we actually have a lot more questions sort of rolling in at the very last minute. I think we'll keep track of those and try to get back individually. I think we have the names associated with those. So if there are any other questions outside of this, please contact me or Xiaoyu at the emails above, and we can potentially relay questions to Jean-Pierre as well. So with that, I would like to thank our two speakers. Thanks for your time and your input here. We appreciate it and look forward to working with you in the future.